Hello, good afternoon. Hello, good afternoon. Hello, good afternoon. Can you hear me? Hello, good afternoon. For those that have already joined, can you please indicate in the chat? Can you hear me, please? Can you hear me? Okay, okay, okay. Thank you. Good evening, good afternoon once again. You are welcome to today's lecture on enzymology. Today we are going to be discussing about enzymes and what enzymes are, what do they do, and what is their biological role in the system. So enzymes, they are biological catalysts, and we can say that they play a vital role in catalyzing reaction in our body, and we can say that uh, they are mostly proteins, with the exception of some ribonucleic acid enzymes that are called ribozymes. All enzymes are proteinous in nature. So enzymes as catalysts of biological system, although the phenomenon of fermentation and digestion had long been known, the first clear recognition of an enzyme was made by Payen and Pessoz when they found that an alcohol precipitate of malt extract contained a heat labile substance that converted starch to sugar. And this substance, which is now known as amylase, was at that time named diastase. The first enzyme to be obtained in a pure form was urease, which was crystallized from jack beans in 1926. And the work of this scientist, James Sommer, for which he was awarded a Nobel Prize in 1946. Since then, there are thousands of different enzymes that have been purified, and their structures are worked out. So with the exception of few catalytic RNA molecules, which we call ribozymes, all enzymes are proteins in nature. They range in size from large multiple subunit complexes, which are called multimeric enzymes, to small single subunit forms. And the region within, there is a particular region within the enzyme molecule that contains some chemical or functional groups to which the reactants, which in enzymology we'll call the reactants as substrates, they bind, and is called that particular region where reactant or substrate binds in the enzyme. It's called the active site, or otherwise known as catalytic or substrate binding site. So there is also a particular arrangement of an enzyme's amino acid side chains in the active site, and it determines the type of molecules that can bind and react there, because the amino acids that are at the active site, the type of functional group they contain determine which type of molecule is going to bind to the active site. So the active site of an enzyme is usually found on a place that is called cleft or pocket, and this place is lined by amino acid residues that participate in the recognition of the reactant, or what we call substrate. So if we look at this figure, we can see that this is a representation of an enzyme with the active site. If you look at this gray shape, okay, this shape in gray, we can assume that this is our enzyme, and this is the active or binding site where our substrate here can come and bind. It has a shape that can fit and bind to the enzyme. A product is formed. So we have the enzyme, the substrate here, combined together to form enzyme substrate complex, okay, and we have enzyme products complex before the enzyme now catalyzes the reaction to produce the substrate, and the substrate are released from the or is released if it is one. If it is two, the substrates are now converted to products. The products is or are released from the enzyme. We have our now enzyme and our product. So this is a quick and a question. With the exception of the dash, all enzymes are proteins. So apart from uh, proteinous enzymes, with the exception of what type of molecules, that we can say all enzymes are proteins in nature. So can you tell us the answer? Just write the answer in the comment section. So let's look at what we call a cofactor or cofactors. Okay, so what are cofactors? We have many enzymes that have small non-protein molecules that is or are associated with or near the active site that determine the substrate specificity. These they are called cofactors, and these cofactors can be either a coenzyme, a prosthetic group, or a metal ion activator. So a cofactor can be a coenzyme. And what is a coenzyme? A coenzyme is a non-protein organic substance which is loosely attached to the protein part. And coenzymes are often derivatives of vitamins. So we can say where we get coenzymes in our diet, we get them from foods that contains vitamins like the vegetables and fruits and so on. So we have vitamins that provide these coenzymes. They may or may not be modified in the reaction. So they can be, these coenzymes can be modified in the reaction, or they may not be modified in the reaction. So those that are altered or modified in the reaction, they are called co-substrates. For example, vitamin B complex. Okay, all the B vitamins, they supply important components of numerous coenzymes. So we are going to see in a short while examples of these vitamins and the coenzymes they provided. So we have a prosthetic group. A prosthetic group is an organic substance which is tightly bound either covalently or non-covalently to the protein portion. For example, we have flavin coenzymes and pyridoxal phosphate and also biotin. They are all prosthetic group. They are what? Prosthetic group. So we have a metal ion activator. The third category of cofactor, we have metal ion activator. This include most divalent ions and as well as other ions, e.g. copper, zinc, magnesium, calcium, and so on. So enzymes which requires a metal ion for activity are called metal activated enzymes. And metal ions are sometimes, when they are tightly bound to the protein portion, they are the enzyme is said to be called metalloenzyme. So enzyme containing such tightly bound uh, metal ion in the protein, we call them metalloenzymes. So the protein portion is called, for such enzymes, the portion that contains only the protein is called apoenzyme, while the entire active complex is called holoenzyme. So we can say apoenzymes together with the cofactor, that is to say the enzyme and the cofactor together, we have what we call holoenzyme. So these are some examples of coenzymes, metal ions activator, and the non-vitamin coenzyme. So for coenzymes, we have uh, uh, biotin, and biotin, the vitamin is what? Biotin, which is vitamin B7, and the reaction it mediates is carboxylation, okay, transfer of carbon CO2 molecule. So we have coenzyme cobalamin, which is vitamin B12, and this it catalyzes alkylation. We have coenzyme A, which is gotten from uh, vitamin B5, which is pantothenate, and it catalyzes acyl group transfer. Acyl group transfer. We have flavin coenzymes, which is gotten from riboflavin, which is vitamin B2, and it mediates oxidation reduction reaction, that is redox reaction. We have nicotinamide coenzymes, which we get from nicotinamide, and this nicotinamide is what is gotten from niacin, which is vitamin B3. We have the pyridoxal phosphate, which we get from pyridoxine, which is vitamin B6, and the reaction it mediates is amino group transfer. So also nicotinamide is oxidation reduction reaction, redox reaction. So we have tetrahydrofolate, which is gotten from folic acid, and folic acid is vitamin B9, and this is one carbon transfer, one carbon group transfer. That is the reaction they mediate. So thiamine pyrophosphate, which is gotten from thiamine, which is vitamin B1, it mediates carbonyl group transfer. So we have also metal ions here, magnesium ion here, it's ATP dependent reactions, zinc ion, carboxypeptidase, alcohol dehydrogenases, 2-ion, oxidases and hydrolysis, non-coenzymes, vitamin, coenzyme, reaction mediated, and ATP, which is phosphate group transfer. We have coenzyme Q, oxidation reduction transfer, and so on. So And after hearing what cofactors are, let's quickly look at how these enzymes are named and how are they classified. So, except for some of the earliest studied enzymes like the pepsin, renin, and trypsin, most other enzymes are known by their common names, which is obtained by adding the suffix ase to the name of the substrate or to the reaction they catalyze. For example, glucose oxidase and transaminases, etc. So we have a systematic scheme for naming or classifying enzymes, which was adopted in 1972 by International Union of Biochemistry, which is now named as International Union of Biochemistry and Molecular Biology. So in this system, each enzyme has a unique name and a code that is used to reflect the type of reaction catalyzed by the enzyme and the substrates that are involved. So this code is a 4-digit number which indicate the class. The first number indicates the class, the second number indicates subclass, the third number indicates subclass, and the fourth number indicates the serial number of that particular enzyme in the subclass. For example, enzymes that are commonly called hexokinase, it has the IUB designation is ATP dehexosphosphortransferase. So this is the IUB designation of this hexokinase. But the enzyme commission number is 2.7.1.1. It means the first number, it means the class of the enzyme, which we are going to see the 6 classes of the enzymes. But for the sake of this explanation, this class 2, they are called transferases. And the second number, which is 7, is in subclass 7, which subclass 7 for this particular group of class of enzyme, they involve transfer of a phosphoryl group. And the subclass 1, which means an alcohol, is the word phosphoryl acceptor. And the last digit indicate that the number is the first, which is number 1. It means the enzyme is the first in the subclass. So we have hexokinase is named as ATP dehexose phosphotransferase with enzyme commission number 2.7.1.1. So it identifies this enzyme as number 1, a member of class 2, which are transferases, subclass 7, which involves reaction transfer of phosphoryl group, and subclass 1, which indicates alcohol is the phosphoryl acceptor, and serial number 1, which indicates is subclass. So let's quickly look at the general properties of enzymes. But before we continue, I want to be sure whether the participants clearly. If you can hear me, kindly chat kindly send a message in the chat so that I can know that we can proceed. Okay, let's proceed. So let's look at the general properties of enzymes. Enzymes possess enormous catalytic power. They have this tremendous catalytic power for reactions that are not aided by enzymes. The reaction tends to be slower than the reaction that are involved by enzyme, that the reaction that enzymes are involved. So most accelerated reactions by factors of at least 1 million. So when we consider a reaction catalyzed by carbonic anhydrase, and this carbonic anhydrase is the enzyme that convert the combustion of carbon dioxide and water to carbonic acid. So this enzyme is responsible for the effecting the transfer of carbon dioxide from tissues into the blood and then to the alveoli. So each enzyme is capable of hydrating 10 raised to power 5 molecules of carbon dioxide in 1 second. So the catalyzed reaction is 10 raised to power 7 times the times faster than the uncatalyzed reaction. So the enzyme is able to hydrate 10 raised to power 5 molecules of carbon dioxide in 1 second, and also it's catalyzed the reaction 10 raised to power 7 times faster than the uncatalyzed reaction. So they are highly specific in both the reaction catalyzed and their choice of reactant. 4 distinct types of enzyme specificity have been identified. So when we say these enzymes are highly specific for the reaction they catalyze, and we can give example of these types of specificity. We have a linkage, absolute specificity, where the enzyme catalyzes only one reaction, and we have group specificity, in which the enzyme acts only on molecules with specific functional groups. And we have linkage specificity. In linkage specificity, the enzyme acts on a particular type of chemical bond, regardless of the rest of the molecule. So when you look at trypsin, it splits peptide bonds on the carboxyl side that has lysine and arginine residues, while thrombin acts only on the side chain on the carboxyl side when there is arginine, okay, and the on the one of the amino side is what? Glycine. So for thrombin, it acts only when the side chain on the carboxyl side is arginine, okay, while the one on the amino side is glycine. So that's when thrombin will act, okay. So we have stereochemical specificity, and for this type of specificity, the enzyme acts on a particular steric or optical isomer. So the third property is that these enzymes, they do not alter equilibrium. So let me be clear with you here. Let me go back. Let's enormous catalytic power, and we talk about that. This second one is that enzymes are highly specific in the reaction they catalyze, and under this we talk about the specificity, 4 different types of specificity: the absolute, group, linkage, and also stereochemical specificity. So then we proceed with the third property, they do not alter reaction equilibrium. The combination of substrate and enzyme creates a new reaction pathway whose uh, transition state energy is lower than it will be if the reaction were taking place in the absence of an enzyme. So it means they do not alter reaction equilibrium, but rather the activation energy is lower. And this activation energy is the minimum energy that is required for the substrate to be converted to product by the enzyme. So enzymes find a pathway or a route to which that they lower the activation energy, but the equilibrium do not change. So however, in this process, both the forward and reverse reaction are accelerated by precisely the same factor. So thus, enzymes accelerate the attainment of equilibrium but do not shift their position. So if we look at this figure here, when we have the energy that is plotted on the y-axis and the reaction coordinate on the x-axis, and we have our reactant, as in the case of reaction catalyzed by carbonic anhydrase, we can see that the carbon dioxide and water here, as the reactant is very high without the enzyme. But with the enzyme carbonic anhydrase, you have the enzyme able to find a way, a pathway, or a route that utilizes role reaction. We have reducing any activation energy, okay, and also there's going to be what release of energy during the reaction. So the enzyme stabilizes the reaction transition state, reducing the energy needed to form the products, and enzymes are stable over only a slight range of temperature. So the fourth characteristic or property, um, we can say that generally a higher temperature brings about an increase in the movement of enzyme and substrate molecules, resulting in more collision between the molecules and the formation of product. So a 10 degrees centigrade or Celsius, 10 degrees Celsius rise in temperature increases the activation energy of the molecules by about 12 kilocalorie per mole. However, if the temperature rises beyond a certain point, which differs for each enzyme, the enzyme activity levels it levels out and then decline rapidly. This is because the enzyme is denatured by heat. So because these enzymes are proteins in nature, and you know the proteins they have their level of structure, the three-dimensional structure, this is the tertiary structure, and it enables them to function for a particular reaction or for a particular biological activity that the protein is designated to perform. So when they have their temperature that they work best for, since we are talking about enzymes, they have what we call optimum temperature. This optimum temperature is the temperature at which this enzyme works best. So if that optimum temperature is attained by that enzyme, that is when the enzyme will work best. When you exceed this, when you increase this temperature, okay, when you increase this temperature, you are making the enzyme to lose its what biological activity. Three-dimensional structure is going to be lost because the enzyme is going to be what denatured as a result of what high temperature beyond the certain point that the enzyme can withstand. So for mammals and other homeothermic organisms, changes in reaction rates with temperature assume a physiological importance only in circumstances as fever or hypothermia. There are some enzymes that work best at usually enzymes because living organisms uh, have reactions taking place, metabolism, and this metabolism is catalyzed by enzymes. Okay, metabolism is catalyzed by so many enzymes, different enzymes. So these enzymes for each organism, it works best at that temperature or condition in which the organism uh, thrive or live. Okay, there are some certain bacteria that live in extremely harsh, extremely hot temperature. For example, Thermus aquaticus. Okay, the Thermus aquaticus from which Taq polymerase, and this Taq polymerase is an enzyme that is used in PCR, polymerase chain reaction. This enzyme was obtained from that Thermus aquaticus, and this enzyme can work at a temperature as high as or almost 90 degrees Celsius. Okay, a temperature where all enzymes that are contained within human body cannot work. You see, so all organisms have their own environment, and the enzymes in the organisms tend to work best at a defined uh, 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 temperature and or pH, as we are going to see in later on. So they have optimum condition. For all enzymes have optimum condition of temperature, condition of ionic strength, condition of pH, okay, substrate concentration, okay. These are all conditions that will warrant whether the enzyme will work optimally or not. So the fifth property is that enzymes are stable over only a limited range of pH. So each enzyme has an optimal pH that helps maintain its normal configuration in the environment where it operates. So outside this range, changes in the charges on ionizable amino acid residues result in modification of the tertiary structure of the protein and eventually leads to denaturation. For example, the pH optima include those of digestive enzymes, pepsin, which is the pH is 2.0, for trypsin, the pH is 10.0, and chymotrypsin, the pH is 8.0. So here we can see that these are digestive enzymes. Okay, pepsin works at acidic, extremely low pH, which is down to 7, which is neutral, down to 14, which is highly acidic. From 0 to 7, between less than 7, we have acidic, 7 neutral, and above 7 to 14, we have what? Alkaline, okay, acidity, alkalinity, pH. So you see, for the same digestive enzyme, different digestive enzyme, we have pepsin working at extremely low pH. That is the best pH for the enzyme to work. We have trypsin that is working at alkaline pH, and also chymotrypsin, it's at slightly alkaline pH. So if we look at this chart, this figure, it explains the effect of pH on the reaction rate. When we look at the the reaction here, when we look at the action rate of enzyme activity versus pH, we can see that the optimum pH is this point where the enzyme, this particular enzyme pH, the enzyme activity tends to be rising slowly as the pH is increasing. Okay, the enzyme attain its maximum activity. So that is the optimum pH for that particular enzyme. Beyond that pH, the activity declines down to 0. Okay. So the sixth property is that enzymes are influenced by the concentration of substrate. So when we look at the effect of now substrate concentration on the activity of the enzyme, we can say that at very low substrate concentration, collision between enzyme and substrate are infrequent. They do not occur frequently, and the reaction proceeds slowly. So as you increase the substrate concentration, reaction rate tends to increase proportionately as collision become more frequent. So we can say the rate of reaction is proportional to the substrate concentration. When the enzyme begins to approach the maximum rate at which it can combine with the substrate, and the effect of increasing substrate diminishes. So as you are increasing initially at very low substrate concentration, the collision between the enzyme and the substrate is not frequent, but when you are increasing it, you have more collision between the enzyme and the substrate. It means the enzyme is able to collide with the substrate and catalyze the reaction, collide and catalyze the reaction. So as you are increasing more substrate, it reaches a point where the enzyme will be what? Saturated, and the effect of increasing substrates so that you have more collision and more product is going to be formed is going to be diminished because this enzyme is saturated. So we have this saturation point, and the reaction at this point is called maximum velocity, and the substrate at which we have the Vmax, we call it what a Km. Okay, so, and this Km is what Michaelis Menten constant for a particular enzyme. For each enzyme has its own Michaelis Menten constant. So let's look at this chart that explains the rate of reaction versus substrate concentration. When you have rate of reaction at plotted at y-axis, and you have substrate concentration as x-axis, when you will reach a point X where we call point of saturation, whereby increase in the substrate concentration does not really affect the the uh, uh, the does not really affect the reaction rate. So we have this figure as what saturation curve for an enzyme reaction, showing the relation between substrate concentration and rate of enzyme catalyzed reaction. So finally, what we are going to be looking at, we are going to be looking at the classes of enzymes. There are 6 main classes of enzymes. Okay, so although there are some that may define the classes as 7, okay, but the main classes are 6. The classification of enzyme, main classes are 6 classes of enzyme. So we have the first class as oxidoreductases. The second class is transferases. Third class, hydrolysis. Fourth class, we have lyases or synthesis. And fifth class, we have isomerases. And the sixth class as ligases or synthesis. 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 There's difference between uh, synthesis and what? Synthesis. Okay. So, and the seventh class in some of the classification, they added translocases, but these 6 are the main classification. So let's quickly look at each class of the enzyme and explain. So we have oxidoreductases. What they do, they catalyze oxidation reduction reaction. They catalyze redox reaction. So they this includes pair of donors and acceptors, such as saturated and unsaturated carbon-carbon bonds. We have alcohol aldehydes and aldehyde acids and amines and imines. For example, we have alcohol dehydrogenase that catalyzes the oxidation of an alcohol to an aldehyde by the removal of 2 electrons and 2 hydrogen atoms. And subclasses include oxidases, oxygenases, peroxidases, and when we look at oxidases, it transfers 2 electrons from a donor to an oxygen molecule, resulting in the formation of hydrogen peroxide. For example, glucose oxidase, which we have glucose plus oxygen to form gluconolactone plus hydrogen peroxide. This is catalyzed by glucose oxidase. And another one, we have oxygenases, which catalyzes the incorporation of oxygen into substrate, e.g. monooxygenases catalyze formation of a hydroxyl group, and dioxygenases incorporate both atoms of oxygen into a substrate. We have cytochrome P450, P450, cytochrome P450. They are important group of enzymes that uses oxygen in the metabolism of xenobiotics such as drugs and toxins. We have peroxidases, which utilizes hydrogen peroxide rather than oxygen as the oxidant. E.g., we have catalase, which utilizes hydrogen peroxide as both donor and acceptor, and it functions in cell to detoxify hydrogen peroxide. So we have hydrogen peroxide being converted uh, to oxygen and water. This is catalyzed by catalase. So we have the second class, transferases. A member of this class, they catalyze a chemical group from one molecule to another. Thus, they have 2 substrate and 2 products. The major group transferred include amino group, acyl group, phosphate, and glycosyl group. And the subclasses include amino transferases. We have kinases, glycosyl and phosphoryl transferases, and phosphomutases. So for the subclasses, we have aminotransferases, they transfer amino group. For the kinases, they transfer ATP, they transfer ATP molecule, and within the reaction, we have glycosyl, which transfer glycosyl and phosphoryl transferases. Glycosyl transferases transfer sugar unit. Phosphoryl transferases transfer phosphoryl group, and phosphomutases uh, catalyze the conversion of it's a stereo conversion, okay, let's say from L stereoisomer to D stereoisomer. So whenever you hear mutasis, is the rearrangement. There is going to be rearrangement maybe from a particular isomer to another isomer. So for aminotransferases, it's transfer of an amino group from one amino acid to an alpha keto acid, which is the acceptor, resulting in the formation of a few or a new amino acid and a new keto acid. So we have glutamic acid and pyruvic acid. We have alpha ketoglutarate acid and L-alanine. We have hydrolysis, and these are enzymes that catalyze reaction involving hydrolysis, that is to say addition of water to cleave chemical bond. Example of such bonds that cleave carbon oxygen, carbon nitrogen, oxygen phosphorus, carbon sulfur group. Subclasses include esterases, phosphatases, and peptidases. We have the next class number 4 is lyasis, the catalyzed reaction in which group are either removed to form a double bond or are added to a double bond. And the subclasses include the carboxylases and the hydratases. Some lyases require a cofactor, pyridoxal phosphate as the cofactor. An example is fumarase. This fumarase is an enzyme that is in the citric acid cycle. The fifth class number 5, isomerases. These are enzymes in which are involved in moving a group or a double bond within the same molecule. So they involve moving a group or double bond within the same molecule. Okay. So this include cis and trans is isomerases. These are isomer catalyzing enzymes. They catalyze reaction that involve changing of one isomer to another one. So we have cis and trans. For cis, we know cis and trans configuration. Uh, uh, so these are what isomers. We have cis and trans configuration, right? And we have aldose and ketose transformation. So these are examples of the reaction that isomerases catalyzes. Aldehyde functional group to ketone functional group, aldose to ketose. The enzyme is called a mutase when a phosphate is moved from one carbon to another, e.g. conversion of 2-phosphoglycerate to 3-phosphoglycerate by phosphoglycerate mutase. So when the reaction involves changing of phosphate group from one carbon to another, it is what mutase. So epimerasis and racimasis changes the stereochemistry at a carbon atom. The conversion of D and L isomer from, let's say, from D lactate to L lactate is catalyzed by a racemase, while the conversion of D-xylulose 5-phosphate to D-ribulose 5-phosphate is brought about by an epimerase. So the sixth class, they are called ligases, and these are enzymes that catalyze bond formation between two substrate molecules, and energy is required for the reaction to occur. Typically, the energy is supplied by ATP, and when this ATP is hydrolyzed, it moves the reaction forward. So they are called synthetases. Whenever you hear synthetases, it means the reaction utilizes hydrolysis of ATP. For example, to add carbon dioxide to pyruvate, carbon dioxide is going to be incorporated into the coenzyme biotin, which requires hydrolysis of ATP, and the carbon dioxide is transferred to pyruvate by the same enzyme. So let's look at this table, which explains the major classes of the enzymes, and with the first enzyme commission integer. You remember the enzyme commission number is 4 digits. The first digit means class of the enzyme, and the second digit means subclass, the third digit means subclass, and the fourth digit means the serial number of that particular enzyme in the subclass. So the first integer, we have oxidoreductases, which carry number 1, and the type of reaction is oxidation reduction reaction. Number 2, we have transferases, chemical group transfer. Let's say from AX, when you have AX, A is being removed and X is being transferred to B. We have A plus BX. Hydrolysis, hydrolytic cleavage, they are number 3. The fourth class, lysis, non-hydrolytic cleavage, leaving double bonds or addition of groups or to a double bond. And the fifth class, they are isomerases, change of geometric arrangement of a molecule. And the sixth class, they are what? Ligases joining together of two molecules accompanied by ATP hydrolysis. So let's quickly answer this question in the chat box. List 3 alias studied enzymes. Please list 3 alias studied enzymes. You can post the answer in the chat box. So this brings us to the end of one. We are going to look at other things later on. So I believe you can have 1 or 2 questions to ask. If you have any question, you can drop it in the chat box so that we can respond and get more clarity. Any question? Any question? Okay, you can always drop the question in the chat box so that we can always come back and attend to it. Thank you very much for being here with us. See you next in our next lecture. Bye bye.